so I don't forget to do the recording, of course. All right, so good morning on the recording, and I'm just going to quickly review the hormones that we've actually discussed so far. Let's get a list going. Lists are always good. Lists are basically flowcharts in their own way. So let me share my screen. Share, share, share. All right, so you guys tell me, what have we discussed so far? What was the first major hormone that we actually started to look at in terms? I know we've kind of been all over the place, and that's not a bad thing. Good. Growth hormone releasing hormone. I like that you went there. But of course, growth hormone releasing hormone helps with the release of growth hormone. So as a list goes, we can start writing these things down. So I got growth hormone. And then when I write this down, I can do exactly what Tariq did, and I can say what hormones are associated with this, and I got G, H, R, H. And then if you recall, there's actually a hormone out there that will inhibit growth hormone as well, and that's growth hormone inhibiting hormone. So when growth hormone gets too high, what hormone is going to help bring growth hormone levels back down? What do we know? This guy here is also known as somatostatin. And you guys will see somatostatin later in the semester. Anytime somatostatin is in the system, it's going to try to slow down growth. Slow down growth. We're good. We don't need to grow that much more. We'll be good. Somatostatin, GHIH. All right, somebody else give me another hormone that we've discussed so far, please. T3, T4, love it. Thyroid hormone is often just written as T3, T4 or thyroxin. And if we look at thyroxin or T3, T4, we know that there is TRH, and that's going to cause the release of TSH, and that's going to cause the release of thyroid hormone. Great. CRH, which of course is going to be associated with what major hormone? CRH downstream, what major hormone are we thinking about when we start thinking about CRH? Cortisol. So we got cortisol, our stress hormone, quote unquote, right? And that's all about CRH, which causes the release of what from the anterior pituitary? Good. ACTH, adrenocorticotropin hormone. Good. And that's going to cause a release of cortisol. All right. What else do we got out there? We have talked about another one, whether you fully remember it or not. TRH is right here. Ashley, good job. Yep. TRH, TSH, T3, T4. Good. Insulin, aldosterone. Thank you, Ruju. Yeah, glucagon. Gosh, these are all great ones. Let's go ahead and write these down. We haven't fully discussed insulin and glucagon yet. So let's go ahead and write aldosterone first. Now, aldosterone is a bit of a tricky business because aldosterone is not through this anterior pituitary, the hypothalamic anterior pituitary axis. What did we learn is the function of aldosterone? You guys might need to look back at your notes, or I'll just have to remind you. Helps regulate minerals. And which two major minerals is it regulating? Yeah, it's acting on, actually not glucose. Long term, it will have an impact on glucose, Tariq, but this isn't about glucose, strange as it may seem, because all the rest are. This is about sodium and potassium. What does it do to sodium? Does it help us keep more sodium, or does it cause us to release sodium? Anybody remember that? Good, keep, right? So this is sodium retention. Sodium retention And what about potassium? Is it doing the same thing? Is it retaining potassium, or is it going the opposite direction? Yeah, potassium out. So this is potassium excretion. Yeah, good. And the reason it can do this, of course, is because we have sodium-potassium pumps that are moving these two things in opposite directions. We'll learn more about that in the renal system. This is all about salt regulation, and it's really darn important. Aldosterone has other functions like vasoconstriction and increasing blood pressure. So we'll definitely be diving back into aldosterone and multiple times this semester. Pretty good one to cover. Insulin and glucagon. So let's go ahead and write glucagon first. We really haven't covered this in detail, like in terms of its function. Glucagon and insulin. All we've talked about here with these two is where they are secreted from. So who remembers where they're secreted from? Good, pancreas. More specific. Does anybody remember the specifics of these? GFR is not, it's not accurate on this one, Tariq. I love that you went there though. GFR, what you've learned about GFR so far is that's the adrenal cortex, but we're going to the pancreas. Good, alpha cells and beta cells, great. So glucagon is secreted from alpha cells of pancreas. We'll end up seeing what its stimulus is and its function today. And beta cells, those are the ones that secrete insulin. And of course, alpha and beta cells are on the islets of Langerhans. So the pancreatic islet cells. There's one more hormone that we've covered in some detail so far. Actually, in enough detail that you guys should remember it, but it tends to get forgotten. Oh yeah, well, of course, there's those two. So there's two more hormones, really, that we've talked about. Great, good memory. I'm so proud of you. Yeah, seriously. Calcitonin, right? Calcitonin, if we want to look at its function versus its stimulus, right? Calcitonin stimulus was an increase in blood calcium levels. By the way, I don't know if you guys know abbreviations in science yet for some of this stuff. So if I wrote calcium with brackets around it, like this means concentration. So anything in science, if you're looking through a textbook, if you ever see brackets around a word, it tells you it means concentration of. So this is an increase in the concentration of calcium. And then if I write ECF, this means there's an increase in the concentration of calcium outside of the cells. I could have also written plasma, and that would have worked. Do you guys see this abbreviation and understand what it means? Increase in the concentration of calcium, basically in the blood. It's a nice short version, so you can write this down, faster. So the stimulus is an increase in calcium in the ECF, and what's the response going to be? Right? Calcitonin is going to try to decrease calcium in the blood. Does that make sense in terms of the response is compensatory for the stimulus? Yeah, we'll get to adrenaline in just a minute. Good. So calcitonin, there was also PTH, so parathyroid hormone, where the stimulus was a decrease in calcium in the ECF, in the blood primarily, and the response is going to be the opposite because homeostasis, increase in calcium in the ECF. Good memory on those. Sometimes I forget about them too. Yes, good, Ruju, that's exactly right. We want to make sure that we don't forget about epinephrine, norepinephrine. We got to know about these. They tend to be bundled together. You guys are going to see that they aren't exactly the same, but if they are released as neurohormones, we often consider them in the same bag, All right, so norepinephrine, epinephrine, these are secreted by chromaffin cells found inside of the adrenal medulla, and they're all part of a sympathetic nervous system response. So this is sympathetic nervous system, gotta know that. This is an emergency response, and an emergency response only, fight or flight, good. Flight, you got it. And in response, right, the function of these guys ultimately is to increase energy availability. Yes, no? Does that make sense? It's all about making sure that there's enough energy. You guys are actually going to learn how these hormones bind to specific receptors and the exact intracellular mechanism that these things take. You're going to learn about that in the cardiac system. It's going to be running through a, beta adrenergic receptor that's activating GPCRs. It's going to be a lot of fun. Yeah, 6-hour moose chase. You got it, man. See, there's always a link. I'm not just a tangent king. I actually try to associate it sometimes. Most of the time. Sometimes I just straight tangent. So increase energy availability. And I hope you guys are starting to understand this term a little bit better. Availability. Availability. I don't know how to spell it right now. Ability. Ah, geez. Availability. That word just completely went over my head. Anyways, I hope you guys are starting to understand energy availability a little bit better. In that energy is ATP. ATP requires oxygen and glucose. And we can get glucose from lots of different sources, not just glucose sources. True story? We can get glucose from glycerol. We can get glucose from amino acids. And if we're making glucose from glycerol and amino acid, yeah, exactly, Heather, keto. If we're making glucose from glycerol and amino acids, this is called gluconeogenesis. If we're stealing from our glucose stores in our liver, well, glucose is stored in our liver as glycogen, so that would be glycogenolysis. Does that make sense? So when I say increase energy availability, we have actually quite a bit that we can think about with this term. Yes? No? Okay, thanks, guys. Feedback is so helpful. Now, I want you guys to start seeing some links. I've already started to do this for you, but what about growth hormone? What does that do to energy availability? Increase, decrease, no change. Yeah, it increases energy availability. It's a little bit unique in that it increases energy availability and then turns around and uses that energy to the energy away just somewhere else. T3, T4, what does that do to energy availability? Increase, decrease, no change. Yeah, this one's all about making more ATP, and we're going to talk more about T3, T4. So if you guys haven't studied ahead, you might not have that answer in your head. Cortisol, stress, right? What does that do to energy availability? You're thinking long-term, Tariq. Short-term is what I'm after. Cortisol, remember this is essential. This is not, we just associate it with that because it starts to cause the release of glucose, and we want that over the short term. You guys are going to learn that glucagon is all about increasing blood glucose levels. So it's all about allowing for energy availability as well. And then last but not least down here, epinephrine, norepinephrine, energy availability. So look at all of these hormones that we have that help with making energy more available. That's a lot, right? And that's not even everything, estrogen, testosterone, all of these are about increasing the amount of energy available too, and then potentially storing them away. Crazy, huh? Pretend I know how to spell that word. Now, I want you guys to look at this list, and there's one hormone on this list whose sole job is to store energy away. Who am I? One of these things is not like the other. I have one hormone on this list whose sole job is to store energy away. Who am I? I'm going to go ahead and wait until more people answer. Yeah. By process of elimination, that's insulin. Okay. Insulin is all about storing energy away. Increase stored energy. So this is going to store glucose away as glycogen. This is going to store fatty acids away as triglycerides. This is going to build new proteins, so forth, so on. And we have one hormone really that has that function throughout the body. There are a couple of synergistic hormones, but insulin plays the primary role in storing energy away. Sounds pretty important, doesn't it? And that's why when insulin breaks, it's pretty bad. What disease is caused by the breakage of insulin? It's diabetes. And we'll talk more about diabetes today and how bad it is and so forth, so on. So let's start getting back to our actual lecture. I'll just kind of show you guys, here are those islets of Langerhans. We got alpha cells that produce glucagon. We got beta cells that produce insulin. There are actually other cells in here too, gamma cells and stuff, but we won't talk about those. The whole idea behind insulin is that its job in life is to bring blood glucose levels down because it's storing glucose away. So let's go back here and let's do a stimulus and response for this, but I'm going to work backwards just so you guys can understand this better. So if I wrote stimulus, response, we're going to come down here to insulin. What did I just say the job of insulin is? To increase blood glucose levels or to decrease blood glucose levels? Energy storage, but sorry, I want to be more specific here. Increase blood glucose levels or decrease blood glucose levels? What's its job? Insulin, right here, the effect. So we're going to write that down. So the response of insulin is to decrease blood glucose levels. You're going to learn how it does this, but the idea is we take glucose out of the blood and we store it away. That's why the blood glucose levels decrease. Does that make sense to everybody? It's called glucose uptake. So if the response is to decrease blood glucose levels, what's the stimulus going to be? Make sense? Seems like everybody's got this. Right after you've increased your BGL, your body's response is to secrete insulin to bring that BGL back down again. So when's a good time that your BGL starts to increase? What do you guys know about life? When are you going to have a boost in your blood glucose levels? Right, perfect. Not just sugar, Tariq. Food, period. Food, period, is going to be doing this job. There's going to be a lot of reasons why. Don't worry about it. We'll talk more about it in the digestive system, but the primary stimulus for insulin release is an increase in blood glucose levels. There are other things as well that can do it, and there are other things that can inhibit the release of insulin. Like, for instance, if you're running away from Mr. Axeman, do you want to be storing glucose away? Yes or no? No. So what do you think the sympathetic nervous system is going to do to insulin secretion? Activate or inhibit? So just Andy, just real quick here, you guys just got it right in terms of we don't want to be releasing, or sorry, to be storing glucose away when we're running from Mr. Axeman. And when we're running from Mr. Axeman, we're trying to increase energy availability. So the answer would be to inhibit insulin so that we do not store that glucose away. Does that make sense? Andrea, does that make sense? Sorry. Yeah, sorry. And that happens. The way that, sometimes I just talk too fast, so no worries. Perfect. Pure keto will kill you. End of story. I don't even want to get into this argument. Pure keto is called the Atkins diet, and lots of people died back in the 90s due to it. That's why the Atkins diet went to the wayside, and then they started up on the keto now. Ketogenic diet now actually requires, about 20 grams of carbs per day, and that's what's going on there. But that's kind of the idea. Yes, the presence of proteins and amino acids is also a stimulus for insulin release. So if there's amino acids in your stomach, then insulin will be secreted to a lesser degree, but enough to do the job. Good. Yep. Yeah, ketogenic diets are such a fad right now, and people are trying to do them by themselves, and I don't recommend that. I always recommend, if you're going to do a keto, that you get, a regimen from your doctor. Ketos can harm your body for the rest of your life if you don't do them right. And nobody tells people that, right? Because nutritionists just want money. Anywho, going on here. Glucagon and insulin, well, they're opposites. Okay? Glucagon is insulin's nemesis. They're always fighting. Eternal battle for glucose. Forever and always. So what does that tell you? What is the response? What is the effect of glucagon? If it's in a battle with insulin, yeah, glucagon is always trying to increase your blood glucose levels. So what do you guys think the primary stimulus for glucagon release is going to be? Decrease in blood glucose levels. You got it. How do you guys feel about that? It's not too crazy, is it? There's still more to it, of course, but it's great to have a list with as much as you can on it, but as little as possible on it, so that it's possible to actually learn it. You put too much on one page and it just gets out of control. You guys feel okay with this page so far? That these are hormones you've learned, and now you're going to start to dive a little bit more into the mechanics of insulin and thyroid hormone. Those are the two that we're going to focus on for the rest of the semester. And I'm never going to erase it. It's here forever and always, because I don't get rid of my sketch pads. And two, just as a heads up, I'll always give you guys a little bit more time to write it down, of course. But these are all these. Let me show you where, because not everybody knows. But all of these are recorded. And in case you didn't know where the recordings were at, if you go click on Collaborate, this is just for anybody. And there's 3 bars right here. You put your cursor over it, it says menu, you click on that, and then there's a button down here that says recordings, and this is all of the lectures for the semester so far. Yeah, I saw Tariq was uploading to YouTube so that you don't have to have internet access, right? You just basically have to have, your phone service, and you can listen to them or watch them anywhere. So that's pretty awesome. All right. Is that helpful for anybody that maybe didn't know that that existed? I don't know. Anyways, if not, if so, great. Whichever way it goes. Cool. I always find that there's one or two people that don't know that that's possible, so I want to share it. Okay, let's see here. The last couple of hormones that we haven't even written on our list, because we're going to talk about them in a lot more detail at the end of the semester, they are secreted from your gonads. The gonad is the reproductive organ that produces sex cells. So in the male, the gonad is the testis, and in the female, the gonad is the ovary. Males and females actually both produce testosterone and estrogen from the adrenal cortex, but males have more testosterone because they have testes, and females have more estrogen and progesterone because they have ovaries. That's all that I really care for you guys to know at this point, okay? We will cover this in massive detail. You're going to learn all about the histo of this. You're going to learn that interstitial cells are actually called Leydig cells. You're going to learn a whole bunch about this later. Clear as mud? Murky, murky mud. There are a lot of other endocrine organs that we will be discussing this semester. Heart, GI tract, kidneys, all three of these we're going to go in a lot of detail about. Lungs, right? Those actually secrete hormones, enzymes that act as hormones. So yeah, you better believe we're going to be dealing with that. Well, guess what? Always the bridesmaid, never the bride. Liver produces hormones. So we got to make sure that we think about the body as more than just these very strict endocrine glands whose only job in life is to produce hormones. There are a lot of other organs that have a lot of different functions, but one of those functions is to produce hormones to help them communicate. GI tract, yeah, GI tract, gastrointestinal tract. The primary organs of digestive system, in case you didn't know, is basically called the GI tract. The GI tract is a tube that runs all the way from your mouth to your anus with just a couple of sphincters in between. So the digestive system is literally just one big long tube with literally some muscular doors that can open or close. It's pretty crazy. It's pretty crazy. Yeah, we just got one big long tube from mouth to anus. And you got two sphincters in your throat and your esophagus, and then you got some sphincters going in and out of your stomach, and then you got a sphincter at the, well, two sphincters really at the end of the story. How is it endocrine? Oh my God, I'm sorry, I didn't understand your question at all, but man, it's massively endocrine. There are so many hormones secreted by the GI system. Listen, if anybody thinks the digestive system is simple, wait until we get there. Okay, digestive system is very complex. Let's put it this way. If you look up any pharmacological agent that you will be giving a patient in the future, any and all, there's not a single pharmacological agent out there that does not have a side effect on impacting the digestive system. I know, we're totally a donut. But does that make sense? Every drug on the face of the planet, whether it's recreational or pharmaceutical, will impact your digestive system. So that tells you what? A target cell is a target cell because receptors. Tells you a lot about the digestive system, doesn't it? Whether or not it's your brain that's working with your digestive system, or your fat that's working with your digestive system, or your digestive system that's communicating with everything else. Your digestive system is always in communication with everything. Yeah, yeah. And serotonin is literally just, kind of a hunger system thing. And it's got some other impacts. Serotonin's pretty pretty big time hormone, but actually neurotransmitter technically, but can be released as a hormone. But no, there's a lot of other ones. I mean, just look up ghrelin. Ghrelin, the really cool one. Ghrelin, spelled all funky too. But ghrelin is a great one. CCK, cholecystokinin. So you guys are going to be learning about a lot of these. Motilin, GLP-1, GIP. So there's a whole bunch of hormones from the digestive system, and they're all basically trying to communicate with the rest of your body about, guess what? Energy. I have enough fat. Stop feeding me fat. I have enough glucose. Stop feeding me glucose. Talking to insulin, talking to glucagon, talking to epi-norepi. So there's a lot of things going on in your body. All right, cool. Good questions all. Let's go ahead and close this one. Don't save. Now, Endocrine 4 is going to step up your game. You guys, I hope, have your brain ready. What we're going to go into now is more cellular physiology. We need to start talking about how the things work, not just what things are in the basic, hey, there's some energy changes here and there's some energy changes there and there's more cell proliferation here. No. We need to go in and we need to talk about receptors. We need to go in and talk about the movement of ions. We need to go in and talk about the synthesis of new proteins. So we need to dive down into the cellular level because I hate to inform some of you, but anatomy and physiology, medicine is not about organ function. Physiology doesn't happen at the organ level. Physiology happens at the cellular level. Does that make sense? When you give a patient a drug, you're acting at the cellular level. All physiology happens at the cellular level. And it's a bit of a wake-up call, unfortunately. But I promise I'm not going to go too crazy, and I will also make sure to walk you through most of what I want you to know, if not all, hopefully. So we're going to start with the thyroid gland. And here in the thyroid gland, we're going to focus a little bit more on the production of T3 and T4. I have tried to lead you to what you need to know about it as of now. For instance, what does this iodo stand for in both triiodo and tetraiodo? It tells us iodine. So tri, 3 iodines, tetra, 4 iodines. In addition, what classification of hormone is thyroid hormone? Is it a steroid, a biogenic amine, or a protein? Good, it's a biogenic amine. But what is unique about it? What makes it unique? What makes it special? What makes it different than other biogenic amines? Yes! You guys are such good listeners, and I love it. It's nonpolar, which tells us what about it? What does that tell us about it? Is it lipid soluble, water soluble? Yeah, it's lipid soluble. So how does it move around the body? As long as it's in a nonpolar form, how can it move around the body? Needs carrier proteins. Wherever there aren't carrier proteins, it moves by passive diffusion. So those all go hand in hand. So you see how powerful the knowledge of a little bit of chemistry is. So I love that you guys are taking to heart. That makes me super happy. Next, let's go to here. Just quickly draw this out. Let's go ahead and do our flow chart. All right, this is a great template to start writing down and studying each one of these. Thalamus. So I got a hypothalamus. Hypothalamus secretes to the anterior pituitary. And the anterior pituitary is going to secrete down here to a target cell. Target cell is going to have a response. Then, of course, the response is always compensatory. So whatever's going on with this response, we're going to have some sort of negative feedback loop that's going to go back and try to do the opposite of the upper portion. And we always got this stimulus up here as well. Wish I can get rid of this bottom snapshot area without messing everything up. It used to just be able to, oh, that was working. Woohoo, that worked. That wasn't easy, but it worked. All right, so now, beginning of the story. I'm just gonna kind of write this down because I got time that I wanna get through here. But the beginning of the story, of course, is we have ourselves TRH, thyroid releasing hormone, TRH, which is going to move those couple of centimeters down from the hypothalamus through that portal system to the anterior pituitary, where we have receptors for TRH, and the anterior pituitary now secretes TSH in response. TSH, this one's an important one. TSH is going to go down to these follicular cells of the thyroid gland. And the follicular cells, in response to TSH, will start to produce more and release more T3, T4. But the only way that we can release more T3, T4 on demand is if we store it away and release it when we want it. Because otherwise, since this is nonpolar, as we made it, it would go into the bloodstream. Does that make sense? So we can't just make more. We have to store it and then release it from storage when we want to release it from storage. Is that clear to everybody? We did this yesterday too, so hopefully this is a redundancy. Let's never, you don't understand. Okay, we'll do more of this. No worries. Thank you for letting me know. So just do one more tie. What do we know about T3, T4? When the levels of T3, T4 get too high, what's that going to try to do to TRH? Increase, decrease, no change. Let's go back. When T3, T4 levels are too high, it's going to be trying to bring TRH back down because homeostasis, always the opposite effect. Does that make sense? For anybody that might have gotten it backwards? Oh, that's funny. And then what is it going to be doing to TSH? Increase, decrease, no change. Always the opposite, always the opposite, always the opposite. We're going to kind of look at some case studies today on this as well. If we have to talk more about insulin tomorrow, then we'll do it tomorrow, and that'll be fine. Okay, cool. So T3, T4. Let's just do this real quick. And actually, I'll use my scratch for this. If I have a cell, no worries, Heather, no worries. I'll get you, I promise. If I have a cell and it produces a normal biogenic amine, then that biogenic amine is going to have a charge on it, normally a negative charge. So we're going to write a big negative charge on this biogenic amine. Is this clear what I'm doing right here? I have a cell and I have a biogenic amine that's negatively charged. And if I have a biogenic amine that's negatively charged, can it make it through the cell membrane on its own? Yes or no? This is for everybody. If I have a negatively charged anything, whether it's sodium, potassium, bicarb, or whether it's a biogenic amine that's negatively charged or a protein that's large and in charge, can that make it out of that cell membrane all on its own? The answer is no. So if it's a biogenic amine or a protein, then it needs a vesicle. If it's small enough, like an ion, then it can go through a channel. But we don't have channels for this because it's still too big. So it's inside of a vesicle. So in order for this to be released, this biogenic amine that's charged to be released, it's going to need exocytosis for that function. True story? Now here's the deal. It's sitting in a vesicle. It can't get out on its own, so it needs a cell signal. It needs communication from somewhere else to cause this exocytosis to occur. Does that make sense? It can't do it on its own. True story? It needs help from somewhere. It needs communication from somewhere. All right, so now what if I had a biogenic amine that was not charged? Right now, it's not charged and it's not bound to anything. It's not stored on anything or anything. It's just being produced. Everything in the universe is trying to go from an area of high to low or low to high. Everything in the universe is trying to go from high to low. So if I'm making proteins, if I'm making a non-polar biogenic amine inside of my, my cytosol here, as it's being produced, what's happening to the concentration of the biogenic amine inside of the cytosol? Increase, decrease, no change. Did you catch it now, Heather? You're starting to see it, right? Perfect. That's great. So as the concentration were to increase as we synthesized it, it would simply get it, simple diffusion. It would just passively diffuse right out of this cell as it was made. We wouldn't have any control over it at all. It would just right into the blood as it was being produced. This is what happens with cortisol, testosterone, so forth, so on. Exactly. So if we want to control the release of T3, T4, we have to do something about this. And we do something about this by storing it on a protein in the colloid. And that's why we have thyroglobulin, is to store it away, and then we're going to have to stimulate that thyroglobulin when we want it to be released. Cool, no problem, anytime. That's what I'm here for. I hope that helped other people too. Was that helpful to anybody else in terms of even some of the background in diffusion, right? See, that's why we do this. When one person asks a question, it almost always helps other people as well. Okay, let's see. Here, we're here. There are some other things to add to this. For instance, if a follicular cell is going to make thyroid hormone, what did we say it requires? I'm going to go into the system kind of off to the side here. Gots to have iodine. And where do we get iodine from? Our diet. You got it. So we have to consume iodine from our diet. And we're going to go ahead and say iodine, diet, and I'll just put it all together there. Iodine is that mineral, Trish. You got it. Yep, it's coming from the food we eat. Iodine is in soil. This is an important one for everybody to understand, is iodine is in soil. Oops, I didn't want to do, what is that? Text to speak? Go away. I'm going to see if there's something that exists out there. Oh, there is. I figured there would be. So it's in soil, and there are parts of the world that don't have iodine in the soil. So as iodine's in the soil, the plants absorb the iodine. We eat the plants, or animals eat the plants, and then we eat those animals. Make sense? And then we get iodine from the food we eat. Yes, no, maybe so. We don't synthesize it, anything along those lines. This is global experience, legislation for salt iodinization. Where's just a iodine soil deficiency? Oh, they're here. So this is a world map deaths. So I definitely want to point out a couple of places in the world, especially Africa. Africa is very iodine deficient. The soils of a lot of the African world does not have iodine. Crazy, huh? Why did that just do that? I guess I clicked on it. And so these are deaths related to iodine deficiency. Look at where most of the deaths occur. There are other areas like in the Andes Mountains, right? Down here in the west coast of South America. I mean, when you're that high up in elevation and the soil is pretty darn thin, you know, there's not a lot of iodine there. Mongolia, there's a lot of issues with getting iodine to Mongolia. But this is why we started to iodinize our salt. It's to supplement the lack of salt. Sorry, the lack of iodine in food that these people have. Pretty crazy, huh? But we do our best to try to make sure that iodinized salt gets out to the rest of the world. Pretty cool. Now, what happens if there's not enough iodine? So let's come back to this and just remind ourselves that there's not enough iodine in our diet, what's going to happen to the production of T3, T4? Increase, decrease, no change? Good. And this is called, gosh, I did it again. All right, come back here. This is called hypothyroidism. Now, let me just show you hypothyroidism. Hypothyroidism and we're going to end up bringing about, I'm trying to show you somebody with hypothyroidism, but I guess I wrote in the wrong thing. Hypothyroid, yeah, symptoms. No, I didn't do that. Symptoms. I didn't do it either. All right, let's try this. Face symptoms is always fun. All right, so now you're going to actually start to see, maybe not. So there's something called exophthalmos, which is these bulging eyes, and it changes the way that, oh man, yeah, you got your rosy cheeks that go on. You have gaining weight, that sort of stuff that occurs in a hypothyroid patient. So there are distinct, I wish I'd had better pictures for this, but I wasn't prepared there. And another symptom that you often see in these patients is called a goiter. Exactly. They're wonderful. When you're looking through a lot of these pictures, you'll notice that a lot of the people with goiters are of African descent and South American descent, and that's because in these places of the world, the soil is iodine deficient. So what exactly is a goiter? In some places of the world, by the way, goiters are considered beautiful. It's part of their culture. Non-toxic goiter. Man, look at that poor guy with his half-moon face. So do you guys kind of get this idea that the goiter is this swelling in the neck? So what exactly is swelling with the goiter? What do you guys think is actually swelling with a goiter? The thyroid? Yes, no, maybe so? But it goes more than that. It goes more than that. So let's go into this right here, and let's start drawing some arrows around to see what's going on. Yes, it can be reversed if it's iodine related. So if it's just iodine in the diet, guess what you can do? Just start giving them iodine and the swelling will reduce. So iodine in the diet, let's say that decreases. T3, T4 decreases. And now what's the body going to try to do to compensate? What's going to happen to TRH? Increase, decrease, no change. We got an increase in thyroid releasing hormone. Good. What else is going to increase? TSH. We got a problem here. This is thyrotropin releasing hormone. This is thyroid stimulating hormone. And the thyroid stimulating hormone is stimulating follicular cells. Lost iodine in our diet, and all of a sudden those follicular cells are starting to get stimulated. Are they able to produce T3, T4? Yes or no? So what's going to happen to the stimulation of TSH? It's going to continue to increase and increase and increase. So what's going to continue to happen to follicular cell stimulation? Increase, decrease, no change. Oh. And as those follicular cells get stimulated, let's not forget the anatomy of the system. The anatomy of that system is as follows. We have a follicle. And the follicle has all of these follicular cells. True story? You guys kind of remember the anatomy of this using histology? Here's those follicular cells. That's what we're actually describing here, the follicular cells. And then what's in the middle of all of this? Colloid. And what protein is inside of this colloid? Thyroglobulin. Oh. And guess where that thyroglobulin comes from? It's made by the follicular cells too. So as TSH levels increase, we're going to get more thyroglobulin. But if there's no iodine in the diet, will we also get T3, T4? Nope. We continue to stimulate TSH. We continue to stimulate follicular cells. They keep making thyroglobulin. They keep making thyroglobulin. They keep making thyroglobulin. And that follicle is going to swell up like a balloon, a never-ending balloon. Does that make sense? That's why it's called a disease, not normal physiology at this point. It's just the body saying, I'm trying to compensate. This is negative feedback. I'm trying to fix the problem. It's not getting fixed. It's not my fault. I'm doing what I can. I don't know what's going on. Everybody on board with this? Everybody see how that would work? And we're going to draw some flow charts out to make us understand this a little bit better. In order to do that, let's draw one follicular cell, just one. And we're going to draw it as a big old square or a big old cube, I guess, because this is going to be a simple cuboidal cell. So this is going to be a follicular cell. There's one follicular cell. On one side of this follicular cell, out here, we're going to have our blood. So this is ECF out here. So this is going to be our plasma. This is going to be the interstitial fluid right here. This will be ICF here. And then on the other side of the follicular cell will be the colloid. You guys kind of have a representation, even though we're super zoomed in. Do you guys have an idea, or do you want me to show you a picture of what I'm talking about? Sure thing. So this is what I'm talking about. So we can take and probably zoom in on this right here a little bit, although it's not exactly easy. But if I was talking about a single follicular cell, I'd be looking at this follicular cell right here. And there is blood supply out here outside the cell. There's always blood supply. One side of the follicular cell would be facing the blood supply, and the other side of the follicular cell would be facing the colloid. Does that make sense? You'll also be seeing that same thing here, right? This is basically what we're going to end up drawing out. One side of the follicular cell faces blood supply, and the other side of the follicular cell faces the colloid. Okay? Did that make sense? Awesome. Alyssa, did that make sense? Was that pick helpful? Yeah, no problem. No problem at all. Okay, so let's go back here because I find that if too much is on a page, check this out, right? Who looked at this and went, shit, I got to learn that? Dropped your pen. F my life, right? F my life. I hate this. I'm done. It's not as bad as it looks. I promise. We just do one step at a time, and you guys will be okay. It will make sense. We just got to do it one step at a time. Because where do we know T3, T4 is produced from? The follicular cell. What do we know is required? Nah, everybody here is good. Remember, I am aware of how little physiology you will use in your future career. I am very aware of it. So I try just to make it fun, something that you guys can go back to later. You will use it, you just won't realize you're using it because physiology makes up everything that we do. But still, follicular cells we know will make T3, T4. What do we know is required to help make T3, T4? What ion is essential that we get from our diet? Iodine. Now, iodine's a funky little beast because iodine actually isn't in our blood as iodine. Iodine is I2. But because iodine has ionic bond between it, iodine dissociates in water, and we only end up with a single ion, an iodide ion floating through our bloodstream. So I got this right here. I got this little iodide. It is what it is. If you remember iodide, I'd appreciate it. Or you can just write this symbol I with a negative sign. That's floating through your bloodstream right now. And if I'm going to make, nice, if I'm going to make T3, T4, then I need to get this iodide into this cell, don't I? True story? You guys are actually going to find that T3, T4 is made over on in this side, in the colloid, as bizarre as that may seem. So I'm going to need to take this iodide and I'm going to need to get it into this cell. But this cell, surprisingly, has a lot of iodide. So this cell already has quite a bit that it stores inside of it. The iodine levels in here are relatively high. This makes a difference. So we're going to say there is an increase in iodide concentration inside this cell. We got a problem, don't we? Because if iodide is relatively high inside the cell, if I want to move this iodide into this cell, it's going to be moving from low to high. Ah, lovely, Tariq. Great thinking, Alyssa. If I had an open channel here, if I just had an opening, an open channel, a protein channel, everything in the universe is trying to go from high to low. So I can't have that because that would move the iodide out of the cell into the plasma. True story? So in order to move something from low to high, what do I need? Passive transport or active transport? Gotta have active. And what does that tell us? What does all active transport use for its function? We know we're going to need ATP. You don't need to know this exact iodide transporter. I'm not going to teach that to you, but I do want to make sure you understand it's an active process. This is important because as ATP levels change, the iodide transport starts to change, and that can affect people for sure. All right, cool. Well, now that we got an ATP transporter here for iodide, iodide can be actively transported into this cell. And it's going to continuously get pumped out of the blood into the cell, which is going to do what to the concentration of iodide inside this cell? Increase, decrease, no change. Lovely. Perfect. Perfect. Well, cool. What now if I had a channel for iodide on this side of the world? What if I had a channel? What if I had an iodide channel? What can happen to iodide now? If I just have an opening for iodide, it specifically allows iodide to pass through it. Now iodide can move from high to low. So this iodide is going to go from an area of high, and it's going to move into the, where is that iodide going to go? From high to low. So where do we now have a more iodide inside of the colloid? Make sense? Now, what do we know colloid means? Colloid, what is in this solution to make it a colloid? Good. Tariq got it right first time. Proteins. Proteins. Now we got a protein shake. Anybody had one of those protein shakes? Maybe put a little bit too much protein in them. It's like sludge, right? There's not a lot of liquid going on in a protein shake. True story? So there's not a lot of free water. And if we don't have a lot of free water, then things that are, dissociated in water, well, there's not a lot of water, so they're not dissociated anymore. So the iodide that's moving through this cell, where's my gray, because that's what I was using. The iodide that's moving through this cell right here is going to start to reform into iodine because there's not a lot of water. So this right here is going to end up converting into I2. As more iodide enter the colloid, boom, we reionize and we make iodine. So now we have a lot more iodine inside the colloid, boom. Step 1, we just finished it. And if you look at what this has in our lecture, you'll notice I got iodide out here. How did I get it through all of these membranes here? Active transport. Why does iodide diffuse through the cell? Because it's moving from an area of high to low. What sort of transporter would we have over here on the apical surface near the colloid? Well, it's got to be a channel. That iodide now is going to reionize into iodine. Does that make sense? We did it. We showed it again. Redundancy is the key to learning. I hope you guys were following along there. Now, what do we know about thyroid hormone? Is that it's nonpolar. So as long as we have thyroid hormone, this biogenic amine, all by itself, it's going to just diffuse wherever the hell it wants to go, really. So what do we know we need to store this T3, T4 on? What do we know? What's the name of the protein? Not vesicle yet. Go back a little further. What's the name of the protein that we need to store this T3, T4 on? Thyroglobulin, TG. Good. All right, so let's go back and figure out what's going on with thyroglobulin. Well, thyroglobulin is a protein. And to review previous information, we know that proteins, well, shoot, they're encoded for by DNA in the nucleus, aren't they? So if I have a nucleus up here, part of the DNA of the follicular cell, well, its function is to make new thyroglobulin. So we're going to end up with a signal that causes transcription, translation, and we're going to synthesize new thyroglobulin. Does that make sense? TG synthesis. Sweet. We got a small problem here because thyroglobulin is a protein. So as thyroglobulin is synthesized, it's going to have to go to the Golgi apparatus, and thyroglobulin is going to be stored inside of a vesicle, because otherwise these big polar proteins couldn't get through a cell membrane. True story? They're non-permeable. Gotta give them a little bit of help. So now, what if I received a signal, TSH, what if I received a signal that helped to drive exocytosis? So now this vesicle will go down to the cell membrane, fuse, and as it does, it's going to release thyroglobulin into the colloid. Make sense? So now we have an increase in thyroglobulin inside of this colloid. Thyroglobulin is just this little, protein structure. It's like a backbone structure. And I'm going to end up drawing thyroglobulin as just an oval. Pretty simple shape. I'll just draw it as this big, long, skinny oval. Is anybody confused at this point? Because I'm going to keep going with this. We'll finish with thyroid synthesis today, and we'll go and talk about diabetes tomorrow. Is anybody confused to this point by anything? Awesome. So, thank you. Perfect. Thank you for the feedback. I always appreciate it, whether it's yes or no. So, now that we have thyroglobulin and we have iodine, what if I were to take this thyroglobulin? I'm going to take this thyroglobulin here, and I'm going to combine it with some iodine. Well, I could either have a thyroglobulin, long skinny oval, or a thyroglobulin, long skinny oval. By the way, I'm the worst artist on the face of the planet, but that's okay. Now, this thyroglobulin here could either have, in essence, you'll see what I'm kind of trying to draw here. You see how there's these little circles that are bound to it? MIT, DIT, we'll get to that in just a second. Do you guys see those little circles that are bound to this oval in this picture? The oval is thyroglobulin and the circle is the iodine. All right. So we can either have, where are my circles here? We can either have one circle or we can have two circles. And the way this works out is this is called monoiodo. Monoiodo. Mono because there's one, iodo because there's one iodine. And then it's a T because we have thyroglobulin that's attached to it. So this one right here is called MIT. MIT. And this one down here is diiodo. So this one down here is a DI. Whoa, T. I got an MIT and a DIT. And I used to say MIT and DIT. Anybody in here Vietnamese? Or no Vietnamese? No? I had a really funny experience. It was a number of years back, but I'd always be like, all right, everybody in the class, say MIT, 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 so that, kind of redundancy is the key to learning. Say MIT, 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 and the class would be like, MIT, 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 and then I'd say, all right, everybody say DIT, 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 DIT. And I had a group of these Vietnamese girls that all took the class at the same time, and they were just laughing. Just busting out laughing. You know me, you maybe don't know me, but I'm pretty extroverted. So I'm like, oh God, what did I do now? And one of the girls goes, well, dit in Vietnamese stands for fart. So you're just up in front of the class going fart, 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 fart. It's pretty funny. So I got mit and I got dit. Yeah. I'm glad it wasn't something else, right? It could have been anything, but it was just fart over and over and over. MIT and a DIT, mit and dit. Whatever story it takes for you guys to help learn this stuff, right? You got an MIT and a DIT. Now, what about, I got, one second. Just that my neighbor just texted me about something, so I don't know what's going on. So what do we know about T3, T4? How many iodines are in T3? Dumb thing, but helpful. And then what about T4? So what if I took MIT and I bound that MIT to one DIT? How many total iodine am I going to have? How crazy is that? So this is going to be T3. Now this T3 still has the thyroglobulin attached to it. It's still going to be bound to the thyroglobulin because MIT and DIT both have thyroglobulin. Does that make sense? So this still has the TG bound to it. That hasn't changed. What about if I took DIT and I took another DIT and bound it to it? What would I have? T4. And that T4 is still bound to thyroglobulin. Pretty cool. That's how we get it. MIT plus a DIT, T3. DIT plus a DIT, T4. Got lots of farts going on right now. But I got a problem. This has thyroglobulin attached to it, and I need to get it out here to the plasma. So if I'm going to take a protein and get it back into a cell, can it just passively diffuse through the cell membrane? Yes or no? No. No, it can't just get through on its own. What's it going to require? It's going to require endocytosis. So we're going to do the opposite of exocytosis. We're going to now do endocytosis. We're going to take these, T3, still bound to thyroglobulin, T4, still bound to thyroglobulin, and we're going to go through a process of endocytosis. So we're going to take, and these proteins bind to the cell membrane. When they do, it causes the cell membrane to pucker into this vacuole shape, and this moves in this direction, and it's basically going to create a bubble. If you've ever made a bubble by blowing a bubble, you know, you got a flat surface of the soap on that bubble wand, and when you blow on it, it creates the bubble from a flat surface. Yes, no, maybe so? That's literally what's happening right here. And those proteins get stuck inside of that bubble as it's being made. Pretty cool, huh? So as endocytosis occurs, we create a bubble. That bubble's got T3 still bound to thyroglobulin, and T4 still bound to thyroglobulin inside of it. What do we need to get rid of? If I want T3, T4 out here in the plasma, if I want T3, T4 out here, what do I need to get rid of? I need to still get rid of that thyroglobulin. And this is what we have lysosomes for, right? This is what these crazy little lysosomes that you learned about once upon a time and immediately forgot about. You know, they're one of the organelles inside of the body. We have these vesicles that are filled with enzymes. And some of these enzymes, well, their job is to help break things down. And these lysosomes can bind with the vesicles in the body, and as they do, for T3, T4 still bound to thyroglobulin, these lysosomes contain the enzymes required to remove those thyroglobulin from it. So we're going to draw in a lysosome. We'll go ahead and do it as green. Lysosome. We'll do this as green. This is going to merge with this vesicle. And as it does, it's going to cleave the thyroglobulin off of the T3 and the T4. So the TG is going to remove itself, and it's actually going to cycle back. So we can use that TG again, often do. And what's going to be left over? T3, T4. And what do we know about T3, T4? It's nonpolar, so now it can just passively diffuse into the blood. Passive. However, once it's in the blood, can T3, T4 dissolve in your plasma? Yes or no? I know some of you are writing, and that's okay. I'll ask the question again. T3, T4, we've already established, is a non-polar biogenic amine. Therefore, it's basically lipid soluble and it can just passively diffuse through the cell membrane. But once it makes it into the blood, oil and water do not mix. Therefore, T3 and T4 cannot dissolve in the plasma. So what is required to move T3, T4 through the bloodstream? A carrier. A carrier. What is that carrier? It's a carrier called thyroxine, which is T3, T4, binding globulin. Okay, TBG. Thyroxine binding globulin is going to pick up that T3, T4, and it's going to move it to where it needs to go. By the way, what organ of the body makes over 90% of your blood proteins? And it's always the bridesmaid, never the bride. God, that lovely little liver. Don't forget, I like to ask test questions about the liver, and it's always going to be like, you know, this little shock to the system at first, be like, we didn't talk directly about the liver. No, we didn't, because it's always the bridesmaid, never the bride. So if we start thinking about the liver, thyroxine binding globulin. Great question. Binding globulin. Globulins are proteins. They're a very common name for carrier proteins in your bloodstream. You're going to see there's lots of different globulins that we're going to be talking about this semester. Very good. All right. Does everybody feel somewhat comfortable with this flow chart? Let's go back to what the textbook has. Do it one more time, because redundancy is the key to learning. We consume iodine. It dissociates into iodide. Practice? Practice? We talking about practice? Iodide has to move from an area of low to high. So we need an active transporter for that function. As it pumps iodide into the cell, the concentration of iodine is increasing even more inside the cell. So iodide can diffuse down its concentration gradient, pass through a channel for iodine into the colloid, at which point there's less fluid, so we reionize and it becomes iodine. Iodine is going to combine with thyroglobulin. Where did that thyroglobulin come from? Well, this follicular cell synthesized the thyroglobulin, stored it inside of a vesicle, waited for a signal to release it via exocytosis. And now we have thyroglobulin in that colloid. If one iodine combines with a thyroglobulin, we call it MIT, monoiodo. If two iodine combine with thyroglobulin, we call it DIT, diiodo. And now those are going to start to combine with one another. MIT never combines with MIT. MIT can combine with DIT to form T3, or DIT can combine with DIT to form T4. But these are still bound to a protein, not lipid soluble. So in order to get these structures back into the cell, we're going to have to go through an endocytotic event. A new bubble, a new vesicle is formed inside of that cell. Lysosomes cleave the thyroglobulin off or away from the T3, T4, and it enters the bloodstream, at which point it requires a carrier to move it through that bloodstream, and that carrier is TBG. You guys feel like you can go home and practice this? Go home and practice this on your own? That's funny. You're already home, probably. Another one of these lovely flow charts. What is the stimulus for the release of T3, T4? There are all sorts of really cool stimuli for the release of T3, T4. But T3, T4 is all about energy, and as we use energy, we produce heat. Do you guys remember me talking about that a couple of days ago? So anytime we need more energy, whether that's a hypoglycemic state or if it's cold outside, Or if you're pregnant, man, your T3, T4 levels typically go through the roof. High altitude. All of these are in situations that are extremely stimulating for the production of ATP and heat. Why are pregnant women hot all the time? Maybe not all the time, but you get my point. Definitely on the hotter side. Well, that's because of the amount of T3, T4 that's in the system. Lots of these really cool stimuli for T3, T4. Yes, absolutely. For the exam, you can use abbreviations. You better believe it. Absolutely. I'm not trying to kill y'all. For a lab practical, never use abbreviations. Lab practicals are designed, they're literally, can you repeat the word that I taught you? You're welcome. You're welcome. All right. What do we know? TRH stimulates the release of TSH. TSH stimulates the thyroid gland to start to secrete thyroid hormone. Now, this TSH is an important one, as you may notice now. The TSH stimulates the thyroid gland, but remember, it's not just stimulating the release of T3, T4. It's going to be stimulating the endocytotic event. It's going to be stimulating production of new thyroglobulin. It's going to be producing new MIT and DIT. And if it's just thyroglobulin, what did I say about iodine deficiency? If we lose this part of the flow chart, well, the thyroglobulin still exists, doesn't it? Do you guys see how there's two separate flow charts? We are only losing one of these flow charts when we don't have iodine in our diet. We still have thyroglobulin that would be stimulated. Yes, no, maybe so? And that's why, as long as TSH is well, hence not just any disease, but hence the goiter, Tariq. Any disease that causes thyroglobulin to increase inside of the colloid is always going to cause a goiter, regardless of what the disease is. And you're going to find that there's actually quite a few diseases that can cause a goiter. Both hypothyroid and hyperthyroidism can cause a goiter. It just depends on what it does to TSH. We'll come back to that. We'll come back to that tomorrow, of course, because we're pretty much out of time right now. Hypo and hyperthyroidism, there's quite a bit left to talk about with these. We will do a little bit more of this tomorrow because I know people want to know more about why we take T3, T4. You know, basically, if you're hypothyroid, they can give you this synthetic form of T3, T4. Some people take pure T3. Some people take pure T4. Some people take a combination of T3, T4. And the reason why is because only T3 is active. Let me say this again. We're secreting T3, T4. And we combine T3, T4 into this word thyroid hormone. But in reality, the only active portion of thyroid hormone is T3. So if we're secreting lots of T4, which you can see here with this picture, 90% of T4 is secreted. Or sorry, 90% of thyroid hormone is secreted as T4, then 90% of your thyroid hormone is actually inactive. Yes, no, maybe so? So what do we need to do to T4 before our body can use it? Go ahead and answer that in chat. What do we need to do to T4 so that we can use it? Perfect. We're going to have to convert it. Yep, Melissa, exactly. Thyroidectomy, you just need some T3, and that's what they'll do, right? So we got to convert this thing to T3, and your body has a number of different ways to do it. By the way, Melissa, potentially you'll get a combination of T3, T4. It just depends on how functional your liver, kidneys, and the target cells are. So liver is going to be the primary converter of T4 to T3. Kidney also has that function to a lesser degree, and target cells can have that function, but again, to a lesser degree. Liver is pretty cool. If cancer and everything else is okay, I'm guessing they give a combo of T3, T4, to be honest, but I don't know for sure because I don't know how to prescribe, but that would be my guess because everything else is okay. And if everything else is okay, liver, kidney, and target cells are all functioning normally, then you don't need just T3. You don't need to throw an active form into that patient. They can activate it themselves. Does that make sense? To everybody, not just Melissa. Does that make sense? Yep. Levothyroxine. You got it. So levothyroxine, there's a lot of different thyroid hormones that are out there. Synthetic. Synthetic. What's going on with my neighbor here? Synthetic, the thyroid hormones that are out there. But levothyroxine is one of them. And if I remember right, levothyroxine is a combo of both T3, T4, but I could look that up again later. What do I want to do? Oh, so yeah, an easy test question could be back to the liver. Always the bridesmaid, never the bride. What if somebody has liver failure? What's going to happen to their activation of thyroid hormone? Increase, decrease, no change. So now you got a patient presenting with hypothyroidism, but they don't have hypothyroidism, not typical hypothyroidism. They have liver failure. But you still have to treat them the same way. You'd still have to try to treat them with T3 at that point. Less T4, more T3 because their body is having a hard time converting it. Does that make sense? Yep, lab values. You got it. They'd end up looking at all the liver values primarily, but they'd also be looking at T3, T4 levels in the bloodstream and TSH levels. So yeah, you got it. That's why we do so many blood lab results. Did that make sense to everybody though? Like if hypo and hyperthyroidism aren't just about the thyroid gland, there's a lot of other things that can go wrong that can cause similar responses. Now, what if somebody had liver failure and it affected the amount of TBG in the blood? If there was less TBG, what would happen to thyroid hormone transport? Decrease. Now we got another issue, don't we? Because we couldn't just give that patient T3, T4 because it would still have a hard time being transported. So we got to think of another option for that particular patient. Does that make sense? Or we just give them a ton of T3, T4 and then hope that it works. All right, cool. It's 28 after. We'll come back to the effects of T3, T4, and there's quite a few effects of T3, T4. It's a pretty darn important hormone. We'll come back to a couple of disorders, and then we will talk about diabetes, the release of insulin and the function of insulin tomorrow. And we'll definitely get through it all because we don't really have a choice. We don't have a lot of days left to talk about blood. Lab tomorrow, we will be talking about blood. Nothing is due today. Nothing is due today. But what is due tomorrow at midnight? Just to remind you guys, I try to do this as much as possible. Yes, study guide for what? For what? For the cardiac. Yeah, you got it. Perfect. So nothing's due today, but tomorrow the cardiac study guide is due. And then Thursday lecture will continue with our blood discussion, and that will be the end of Unit 1. So you guys will have exams due Sunday, homework due Saturday. I don't want anybody to ever forget. All right, cool. Well, if you have any questions, throw me a text. I hope you guys are enjoying this semester so far. I know it's tough, but hopefully it's not the worst experience of your life. Tomorrow I might be having my my roof worked on, so hopefully it's not too loud. I'll do my best to try to, decrease the ambient noises. All right. Adios.